Hey there, science fans. Bill Nye here, and today we're diving into the fascinating world of recombinant DNA technology. Imagine if we could take genes from one organism and insert them into another, creating new genetic combinations that don't exist in nature. That's exactly what recombinant DNA technology does. Using enzymes and lab techniques, scientists manipulate and combine DNA from different species. It's like taking the blueprints from a cheetah and combining them with those of a house cat, except instead of a super fast house cat, we get amazing things like disease-resistant crops and life-saving medicines. This revolutionary process allows us to create genes with new functions, reshaping medicine, agriculture, and industry. Let's get started. Recombinant DNA, or rDNA for short, is like genetic engineering on a molecular level. Scientists can take genes, those segments of DNA that code for specific traits, from one organism and insert them into another. This creates new genetic combinations that wouldn't occur naturally. For example, bacteria can produce human insulin, a game changer for people with diabetes. That's the power of recombinant DNA technology. How do we get the desired gene into the host organism? That's where vectors come in. Vectors are like delivery vehicles that carry the recombinant DNA into the host cell. Plasmids and modified viruses are common vectors. These vectors are chosen based on the specific experiment and the type of host cell being used. To create recombinant DNA, scientists need molecular tools to cut and paste DNA fragments. That's where restriction enzymes come in. These enzymes act like molecular scissors, cutting DNA at specific recognition sequences. This precision is crucial for creating recombinant DNA molecules with the desired gene inserted in the right place. When restriction enzymes cut DNA, they can create either sticky ends or blunt ends. Sticky ends have short, single-stranded overhangs that can easily pair up with other sticky ends cut by the same restriction enzyme. Blunt ends are straight cuts with no overhangs, but can still be joined together using other enzymes. The type of ends generated by a restriction enzyme plays a role in how DNA fragments are joined together. Once we have our desired DNA fragments with compatible ends, it's time to glue them together. That's where DNA ligase comes in. DNA ligase seals the gaps between the DNA fragments, creating a continuous recombinant DNA molecule. Now that we have our recombinant DNA molecule, it's time to introduce it into a host cell. This process is called transformation. Bacteria are often used as host cells because they're easy to grow and manipulate. The recombinant DNA is introduced into the bacteria through methods like heat shock or electroporation. Once inside the host cell, the recombinant DNA integrates into the host cell's genome and replicates during cell division. This process, called gene cloning, creates multiple identical copies of the desired gene. Sometimes the goal of recombinant DNA technology is to produce large quantities of a specific protein encoded by the desired gene. Scientists use special vectors called expression vectors, which contain regulatory sequences that promote the transcription and translation of the inserted gene. These vectors ensure that the desired protein is produced efficiently in the host cell. Not all host cells will take up the recombinant DNA successfully. To identify the cells that have incorporated the recombinant DNA, scientists use selectable markers. These markers are often antibiotic resistance genes inserted into the vector along with the desired gene. Only the cells that have taken up the recombinant DNA will survive and grow in the presence of the antibiotic. Recombinant DNA technology has revolutionized various fields, from medicine to agriculture and industry. In medicine, it's used to produce human insulin, growth hormone, clotting factors, and vaccines. In agriculture, it's used to develop genetically modified crops. The applications of recombinant DNA technology are vast and continue to expand as scientists explore new possibilities. Recombinant DNA technology is a powerful tool that has the potential to address some of the world's most pressing challenges, from developing new treatments for diseases to creating sustainable solutions for food production. The possibilities are endless. However, with great power comes great responsibility. Ethical debates arise over gene modification. Should we alter human DNA? Despite concerns, this technology continues to revolutionize science. What do you think? Let us know in the comments, and don't forget to like, share, and subscribe.